crystal structure.
Yep. Thought it was so fitting. I was like, this is what, oh shoot. Okay. So I'm just going to be talking about some inhibitors to shikimate kinase. And I read a paper that was published in the Journal of American Chemical Society. I think that's what it is. Yeah. In 2013. So just a review, this is the reaction that shikimate kinase catalyzes. So it's basically just popping on that phosphate group from ADP to this C3 carbon. And so the enzyme has 3 domains, and that are kind of essential to the binding of both ATP and shikimate, and they're important because you have to be able to like know how they bind so that then the potential inhibitor can bind to it as well. And so the 3 domains are the lid domain, which is right here, and it closes over the active site and facilitates the binding of ATP. And the SB domain, which is in green, is right here. And that's where shikimate, the most of the binding of shikimate occurs. And then there's the core domain right here, which is the binding site of ATP, ADP. And so one important thing about this is that the LID domain and the SB domain both have like an open and closed configuration. So when they're bound, the enzyme like kind of like closes over it and just gets like a tighter like configuration towards each other so that then the reaction can occur. And then there's like an open configuration when starting materials can enter and products can leave. And then within this domain, there's like very key specific features that contribute to catalysis. The first is the C4-C5 conformation of the substrate. So the C3 carbon must be in equatorial position for phosphorylation to occur, and C4 and C5 have to be in the axial position. And this conformation is controlled by the enzyme substrate interactions. And then these interactions also lead to causing like the closed conformation of the enzyme. And then the closed lid conformation allows for the activation of the terminal phosphate group, or like the gamma phosphate group on ATP, and it also stabilizes the intermediate of the reaction. And this is stabilized by the guanidinium group on the argine 17 side chain. And then another one is just the closed form of the SB domain, which brings the substrates together, which is, and the closed form of the SB domain is from the certain configuration of the C4, C5 diaxial of shikimate. And then for the shikimate phosphate to be able to leave, the LID and SB domains both need to be able to reopen back up so that the product can be released. So here is just a diagram of shikimate kinase bound to shikimate and ATP. And some of the key interactions you can see are between like this water molecule right here, water 1, and this forms like a bridge between these right here. And then the argine 136 is also an important one, which I will talk about more later because it changes with the different inhibitor, and those just form like the connections to the enzyme between shikimate and the enzyme. And then the ATP ones are right here, and you can see that they all are kind of like, it's just there, and there's like a lot of electrostatic interactions. And then,
so can you go back one sec real quick, Lindsay?
Yeah.
Just to make sure that does everybody see that the shikimate ring is that light yellow?
Oh yeah.
And then the ATP is they're only showing the phosphates, right? Exactly, like she's tracing. So it is kind of like a sweet structure because it's sort of showing you how the groups are oriented relative to each other, promoted by the enzyme, right? Which is part of what the enzyme's job is just to sort of is orientation and then facilitation of the changes that happen during the course of the reaction. So yeah, this is a really nice. Okay, that's all I wanted to say.
Also, I forgot to talk about, you can see like the SB domain and the lid domain, you can see how they're like kind of all a little different. And you can't see very clearly, but I think that's like the slight movement of the two domains and how they get that open and close configuration. You can just kind of see all the different colors, and that's how it like kind of moves. But
yeah.
And so now that when they figured out all of these specific bindings, they then tested inhibitors that were capable of distributing these key structured features and kind of a way to stop or prevent enzymatic activity. Specifically, the inhibitors were made to fix the axial conformation so that they would be recognized by the enzyme and be able to bind, and also to promote electrostatic interactions with the gamma phosphate group of the ATP, because that's also an important interaction to catalysis, and or to reduce the flexibility of the SB lid domain. So then that would basically prevent the enzyme from being able to move, and then therefore it couldn't continue to perform catalysis. Yeah. And then these are just all of the potential inhibitors. I include the Ki values, but I'll talk about that later. And this is kind of how they broke them up. These two both have a C3, C5 ether bridge. This 5 compound, they like didn't even really talk about at all because I just don't think it like really worked. They also didn't really talk about 8. They kind of talked about 6, but the 4 main ones that they focused on that showed like actual potential inhibitory capacity was 3, 4, 7, and 9. But yeah. So how they found.
So can you go back 1 second, Lindsey?
Yeah.
So the bicyclic ones, 3 and 4, and then 7 and 9 have an ammonium group. And that's the position that gets phosphorylated, right?
Yeah.
So they're switching. They're making other changes too, but they're switching the OH that gets phosphorylated to probably in the cell. It's actually an NH3, so it would be basically inert. Okay.
Yeah. And so what they did is they basically just put in the inhibitors with the enzyme, and they actually found that they weren't substrates, which I felt like I was like a little confused about when they were talking about this in the paper, because they said like they found that the inhibitors were not substrates for the enzyme, but then they said when they included shikimate acid that all the inhibitors were able to bind to the enzyme in the presence of shikimate acid, which I didn't really quite understand. And then just to measure the results, they said that they used the PK-LDH assay, which I talked about later. Yes, this week.
I can explain that a little bit if you want.
Okay, yeah.
So the one thing I guess that they wanted to know, first of all, do you all remember that assay that Lindsey talked about? That is the way that they're measuring the enzymatic activity indirectly by measuring the production of ADP, which is like the byproduct after the phosphate group transfer from ATP to shikimate to make shikimate phosphate. But one thing that I think they would want to know is that all of these synthetic molecules, if you add them with ATP to the enzyme, that you don't see production of ADP. And that could happen if the molecule itself, the inhibitor molecule itself, is like a substrate. So imagine that the enzyme is able to transfer the phosphate to that amino group, which is like, could happen, right? If that's the case, then the molecule is not really an inhibitor, it's more like an alternative substrate. You know what I mean? That the enzyme can do a reaction on it. And they're not interested in that. What they want is a molecule that will bind to the enzyme, even in the presence of shikimate, hopefully bind actually preferentially to shikimate, but not undergo any reaction, which is like a definition of an inhibitor, that it instead of shikimate binds, so shikimate doesn't bind, so therefore no phosphate is transferred to shikimate, and you never get shikimate phosphate, and then the pathway to make these aromatic amino acids is stalled, and then the organism suffers.
Yeah.
Yeah.
And then they determined that all of them were reversible competitive inhibitors, which basically just means that when they're there, they inhibit, and then once they're removed, enzymatic activity continues as normal. Okay, so here are the results again, just so you can look at it, and I'm going to talk a little bit more about it. So this molecule here was as I said earlier, had the greatest greatest inhibitory potency with a Ki of 46 micromolar. And so they found that like really none of them were like, they all were inhibitors, but they weren't to the point where they could be used in like a drug because the Ki values need to be closer to 1. But yeah. Okay. I don't know why that is there. So key findings, they found that they all have a higher affinity for the enzyme than the substrate, and they also found that shikimate 3-phosphate is expelled from the active site by 3 arginine phosphate residues. So this is bicycle 3 and 4. This right here is a picture of shikimate kinase binded to shikimate and ATP, and then that's the one in blue, and then the one in this, so the yellow molecule right here, the yellow, goes with the blue, and then the purple goes with the tan. So the purple is shikimate kinase, and then the yellow is the number 3 inhibitor molecule. And there weren't really like that many differences, but one that they did talk about in the essay was this right here. This is argine 136. And they found that there was like just a little bit of difference because of some interactions over here that caused a change over here, which basically just pushed so because of the bridge on molecule number 3, which you can see right here, it just pushed it kind of like out more. So then they just kind of talked about it. They didn't really say it was anything that bad. But then over here, you can see the inhibitor bound to the enzyme, and you can see ADP bound to the enzyme as well. And kind of like Beth, they kind of have like their own kind of pockets that they sit in, which is important for like determining an inhibitor. And then right here, you can see some of the key interactions between right here. I know it's a little blurry, but these are 2 water molecules, and this is argine 136. And then those water molecules and the ether bridge right here in argine 136 forms like a H-bonded salt bridge, which kind of anchors the molecule to the enzyme. Sorry, one second. And they also found that it binds similarly to shikimate, which you can kind of see through the overlay, is that it like basically there's not too many changes, so that they bind in similar ways. But they did find that the interaction favors the SB domain, and sorry, my notes are all out of order. It forms a salt bridge, and but the most important reaction occurs between the water molecule 2013 and the amide nitrogen of leucine 119 and the lipophilic interactions of the C3C5 ether bridge with the carbon side chain. And basically these interactions keep the lid closed. So basically the inhibitor keeps the enzyme closed to disrupt catalysis. And then the fourth inhibitor binds in the same way as the third inhibitor, but the only difference is it doesn't have the double bond, which they found enhances the effects of the salt bridge between the, that keeps the lid closed. And so that's what they think, that's why it makes it more of a potent inhibitor. And also the change of hybridization brings the C3-C5 bridge closer to the carbon side of the arginine 117, which also reduces the flexibility of the side chain, leading to a greater inhibitory potency, which you can see that's, I think this is more right here, and then the carboxylate is like back here.
Mm-hmm.
I think I was talking about a different thing earlier, sorry. Okay. And it just basically, so basically the inhibitors form a very closed, tight part of the enzyme so that then it inhibits the opening and closing and release and uptake of starting materials. And then this is just another summary because I know that was kind of a lot and a little bit confusing. So this is 3, which you can see has the double bond right here. And then this ether bridge does not have the double bond, and they're saying that because the double bond is not there, it enhances the effects because of the change of hybridization. And yeah. Okay, and then, sorry. Gosh. And then, so this is enzyme, this is the inhibitor 9, and that's inhibitor 7. And they kind of categorize them as 3 aminoshikmic acids, and they like added different substituents onto there. They basically just did it with nitrogen. What they found was that you can see that here this has a different they have different stereochemistries compared to the original molecule. And they found that the C3 substituent would interact with the gamma phosphate, causing it to bind similarly to shikimate. And the interaction between the gamma phosphate and the C3 substituent is electrostatic, which basically means it's just van der Waals. And the interaction favors the SB domain and nucleotide remain very close. And there's a lot of H bond residues, which between the inhibitors and the enzyme, which reduces the lid domain and SB domain flexibility. And they actually found that the close conformation of the enzyme with these inhibitor molecules is actually more stable and tighter than when shikimate is bound to it. And they also found that the interaction between the gamma phosphate and C3 is crucial to freezing their binding point to the active site. And yeah. Sorry, I don't know why this is going. There we go. So basically with the 6 hydroshikimic acid 6, they found that it worked terribly. They changed the enzyme's recognition of C1 carboxylate group, causing the movements in alpha helices A2 and A3. And this basically left the enzyme in an open configuration. And then the ligand would undergo a conformational change to the thermodynamically more stable configuration, with C4 and C5 basically in the opposite conformation than what is needed for the closed enzyme to occur. And then the more stable conformation, which doors are more fluid, which means that they move more, and then there's less inhibitory action. And I know that was a lot, but if you guys have any questions, I will do my best.
What does Ki mean?
Ki, so it's, oh my gosh, what is the word? We learned about it in quant. It's K. So it's
is it an equilibrium constant?
Yes, it's an equilibrium constant, but it's with the enzyme parts. So it's the enzyme and the substrate over the enzyme bound to the substrate. And that's Inhibitor, sorry. So the enzyme and the inhibitor as separate entities, and then divided by the enzyme bound to the inhibitor.
So you want a smaller number?
Yeah.
Okay.
So the smaller the number it is, the greater potency it has to inhibit.
Yeah. They write it as 1 over, it's called the sometimes people call it the KD, or the dissociation equilibrium constant, so that the units turn out to be molarity, which is just more comfortable for an equilibrium binding constant. And then, yes, and then the smaller number, the better. So like Lindsey said, all of the numbers that she talked about are micromolar, and having Ki values of like 700 micromolar is like bad. You know what I mean? Like, I always think about it as like, you know how like some medicines you might take like 5 micrograms in this tiny little pill that's mostly like talc or whatever. And then if you take Tylenol, you might take like 2 pills that are pure drug. You might take like a whole gram. Do you know what I mean? So like a Ki of like 700 micromolar means that you would, if you wanted to inhibit shikimate kinase, you would be eating like spoonfuls and spoonfuls and spoonfuls of the compound in order to like affect the enzyme, because it just takes so much to start doing that. Yeah. So the one thing that, if you can you go to slide 12, Lindsey? The one thing, and I think sort of like Naya's paper, actually, since this is the first time people made any inhibitors of shikimate kinase, they're not good, but they're not very potent. And they're also kind of weird looking. I don't know if you think they're weird looking, but the molecules are a little weird looking. And but it was still like the first time, so like I think they're important. And then the other thing that I think is kind of interesting is in that top figure, A with the surface. I think what Lindsey's next paper, just like as a little bit of a preview, they start adding groups that bind that basically closer to to fill the pocket a little closer to the surface. You know what I mean? Because like right now it's like a very small molecule way deep in the back. So they're trying to make it more like an earplug in that thing. Do you know what I mean? Instead of like a gnat going in your ear and nestled against your eardrum, it would be like the whole foam plug. Do you know what I'm saying?